live in about 15 seconds. Hello, everyone. Welcome to this next talk within the preclinical track. In this session, Maxime Lemardi will walk us through the use of PBPK modeling in evaluation and prediction of ophthalmic clinical PK and PD. Before I give floor to Max, a couple of housekeeping remarks. We take your privacy rights seriously. By attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the Q&A tab on your dashboard at any time during the presentation. If you need any technical assistance, please use the chat tab to enter your questions. We'll start this presentation of this session with just a quick poll to see your experience with PBPK modeling. You should all now see the question: how familiar are you with the PBPK modeling? We'll wait maybe 25 seconds to give everyone a chance to respond. I'll wait another 5 seconds before closing the poll. And it is pretty even split. We have about 36% beginners, 40% somewhat familiar with the PBPK modeling, and nearly 25% of expert modelers. So pretty varied audience. Go ahead, Max.
Thank you very much, Vera. And thank you everyone for joining this session today. It is a real pleasure to talk to you in this conference. Thank you for all of the organizers. Vera already said the name of the presentation, which in terms of results will focus on ophthalmic drug products. But before we get there, I just want to give a brief introduction about locally acting drug products and their very specific needs for bioequivalence. So just a few numbers that you're probably already aware, but in the US, the generic drugs, they represent 89% of the prescriptions that are dispensed in pharmacies, but they only represent 27% of the spending paid by customers on insurance. So it is a real generics have a real impact on the cost of medicine, and as the cost reduced, therefore more people have access to those drugs for all of their diseases. So it is very important for societies around the world. I'm not speaking for the FDA, but I've been working with them on some of those projects. So one thing I want to highlight is one of the FDA's mission is to promote generic drug products development. And as we are talking about locally acting drug products, there are some specific issues that we need to address when we talk about bioequivalence. What are locally acting drug products? It is a drug that is going to be used to treat a site of action that is local. It could be the skin, it could be the lungs, the eyes, the GI tract, and any others you may think about. One very specific link for all of the locally acting drug products is that they are not intended to be absorbed into the bloodstream. Therefore, there may not be any detectable concentration in the plasma. And if they are they even linked to what we have at the site of action, which is local? So here on the right, I put just a small diagram with all of the local acting drug products we work with in GastroPlus. So you have the pulmonary, intramuscular, intraarticular, oral cavity, dermal, and ocular. And all of the one with the little stars are the one on which we have active collaboration with the FDA to improve PBPK models, and therefore, in the later stage, impact generic development for those locally acting drug products. I've been talking about bioequivalence, and when we think about bioequivalence, this is the typical diagram we think about. We have two plasma concentration time profile, one for a test, one for a reference. We can calculate the AUCs and the Cmax, and then calculate the 90% confidence interval of the ratio. If this 90% confidence interval falls between the 80 and 125%, therefore, the test and the reference are bioequivalent, and the generic can be approved. This is, of course, a very simplified reasoning. But as I mentioned, for locally acting drug product, we do not have, in most cases, access to plasma concentration to assess the differences between two formulations. And if we do, in most cases, the plasma concentration is not sensitive enough to formulation changes. So we cannot use it as a surrogate to evaluate local concentrations. Therefore, and this is applicable for most locally acting drug products, there are two paths currently used by the FDA based on the drug products and its specific characteristics. On the left, you have what we call the in vitro only option, where you can make a series of in vitro experimentation that compare the test and the reference product. If they have the same Q3 characteristics and performances, therefore, the drug could be deemed comparable and bioequivalent. So that's for the in vitro only option. You can also do in vivo studies, either PK or PD endpoint studies, but those studies are very expensive and require to have a large numbers of subjects, and therefore this is a limitation to drug development. You saw I'm talking about Q1, Q2, and Q3 on this slide. What am I really talking about? Two products will be considered Q1 if they have the same component. They will be Q2 if those components are in the same concentrations within a drug product, and Q3 if they have the same matter arrangement. So another way to look at it is to say that between the test and the reference, they're going to be Q1 if they have the same excipients. They will be Q2 if the ratios of those excipients are the same between both formulations. And finally, if through in vitro testing, we can identify that their physicochemical properties, such as viscosities, particle size, osmolarity, are within 5% from each other, then the drug could also be considered as Q3. Once again, there are some specifics for specific drug products. So based on that general overview for locally acting drug product, I now wish to focus on the ophthalmic route of administrations. Based on the Orange Book, you can see on these slides that for all of the ophthalmic formulations we have on the US market, so emulsions, gels, inserts, ointments, solution, and suspensions, not all of them have generics on the US market. In blue, you have the generics, in purple, the princeps. For solutions and ointments, it seems that most of the drug have a generics on the US market, but for suspension, insert, gel, and emulsions, they are either a very limited number of generics, and those are only for one or two formulations, or no generics at all. Which means that for all of the very complex formulations that are suspension, insert, gel, and emulsions, for which the ability to have the same matter arrangement is difficult to obtain, currently, the current way of assessing bioequivalence for those drug products is limiting the generic development. The development is just too complex or too expensive, and therefore there are no generic drug products on the market for those specific formulations. What is the issue? When we talk about locally acting drug product, as I mentioned, we have a local site of action. Let's say in the case of ocular, for an infection, it could be the conjunctiva. What we wish to obtain between a generic and a drug product is to have the same concentration at the site of actions. If we have the same concentration, we can therefore assume the safety will be comparable between the test and the reference, and also the efficacy will be the same. But in a clinical setting, you cannot really obtain a sample or drug concentration time course in the conjunctiva following the administration of an ophthalmic solution, suspension, and so on. So we need to have other tools to have some sort of a understanding of the distribution of a drug in those tissues. And that tool that has been sponsored by the FDA over the last 10 years is the development of PBPK models to assess drug performances in preclinical and clinical studies. What you see on this screen, and just because of time, I'm not going to give a full description of the model. If you have any questions, I'll be happy to answer any questions inside presentations. But what you see on the screen on the left is the current version of the OCAT model, which is the ocular model in GastroPlus. You can see it contains all of the tissues, all of the ophthalmic tissues, so the aqueous humor, the cornea, and so on. And they're all interconnected to mimic the rabbit and human physiologies. So you have on the right the typical human eye. Of course, this model is defined based on physiological parameters, such as the tissue volumes, the tissue surface area, and then compound-specific parameters, the permeability, and also formulation-specific parameters, such as the particle size, the viscosity, etc. A brief history of this model that was initially created through a collaboration with the industry in 2010, and we had the first version of this model, which was relatively simple. It increased in complexity during the first FDA collaboration that we got in 2014. And since 2014, we had a continuous collaboration with the FDA where we had developed the V3 of this model. And in 2020, so 2 years ago, we got a new FDA grant. And at that stage, the model was pretty much able to describe the PK in rabbit for a lot of formulation, such as suspension and ointments. Where we are right now, which is in 2022, so pretty close to the end of this grant, is going to be next year, where we are trying to extrapolate from rabbit to human and predict the clinical PK for ophthalmic formulations based on PBPK model. And therefore, the idea is if we have a model that is able to predict the clinical exposure once it has been validated using rabbit or other preclinical studies, then maybe we do not need to do clinical studies and we can simply base the generic approval on very well-designed PBPK models and a few additional studies, in vitro or very small in vivo studies. So in the next few slides, I want to present to you one publication we've made where I show how we validated the model in rabbit, and then one example of extrapolation. In both of them, I will talk about dexamethasone. So for all details about the next few slides, you can always go back to the publication I have here on the screen that we published in 2018. And the question at that stage was how a PBPK model can be used to accelerate the development of ophthalmic generic drugs. What we did, and it was somewhat of a general strategy we've been following for all of the case studies we have developed since then, is first to develop the model. And you can see on the screen how the model is able to describe the cornea, conjunctiva, aqueous humor, and plasma exposure following a single administration of Torbalex ST, which is a dexamethasone suspension. Once we had that developed baseline model, we had to validate some of its aspects. One of them was its ability to predict the nonlinear relationship between ocular exposure and the dose. And here we have the aqueous humor observed, Cmax and AUC in red, and the simulated Cmax and AUC in black for 3 dexamethasone suspension that have 3 different doses, 0.01, 0.05, and 0.1. Interestingly, what you can see is that just focusing on the Cmax, when you increase the dose by tenfold between 0.01 and 0.1, the Cmax and the AUC, they only increase by roughly 2.8-fold. So clearly the PK, the ocular PK is not linear, and this is mediated by the ophthalmic physiology. The model, although it doesn't account for any nonlinear processes in its structure, because it's mimicking the ophthalmic physiology, it is able to predict that nonlinear PK for such a suspension. Similarly, we've done validation steps to validate the impact of the suspension viscosity on the exposure, and we found that the model was able to predict the aqueous humor construction time course for 2 formulations with significant differences in viscosities at 70 centipoise for the Torbalex ST and 1.2 for the Torbalex. And by doing that, the model was able to predict relatively well the differences between those 2 formulations. And the last validation set, also for dexamethasone suspension, was to predict the impact of the mean particle size on the exposure. And you can see how the model is able to predict that the cornea exposure is reduced as you increase the diameter of the particle size, which makes sense. A bigger diameter, a smaller dissolution, a slower dissolution, and therefore a smaller exposure. Then we had the questions of using the model for, I would say maybe its first regulatory application. And the question was, can we use the plasma exposure as a surrogate for local exposure for ophthalmic drug products? So we did a parameter sensitivity analysis where we looked at the crossed impact of particle size and viscosity. And then we simply plotted the Cmax and the AUCs for all of those 100 virtual formulations. And you can see that changing the particle size alone have a significant impact, but that is still acceptable in some cases. But when you have viscosity changes, especially in the range of 1 to 40 centipoise, you have clear differences in the Cmax and the AUCs of the cornea and the aqueous humor. So that's for your local tissue that are the site of action. Now, when we look at the plasma, a change in viscosity in the same area, it does have an impact on the plasma Cmax, but it has no impact on the plasma AUC. So that means that we cannot use the plasma as a surrogate for local concentration. And therefore, we need to have other tools. And that was one of the motivations to continue the development of PBPK models to help to the assessment of ophthalmic generics bioequivalence. So another question is, what can we do with a PBPK model? So in this project that we have currently ongoing, we developed a modeling strategy that started with the simplest formulation, the typical solution, and based on PK extrapolation. Then later on, we will focus on PK/PD extrapolation for solution, suspension, etc., etc. I have all case studies for a levofloxacin that I will present tomorrow. So please join that session tomorrow. What I want to present today is a continuation of the dexamethasone case study, which is a suspension. So what we've done is we used the validated model in dexamethasone I've just presented, and we simply changed the physiology between the rabbit and human. We haven't made any other changes. And by doing so, those were the model prediction we were able to obtain. So you're pretty soon going to tell me the fit doesn't look that great, which is true. It seems there is a time change between the observed and simulated profile, especially on the Tmax. We have an estimated Tmax at around 1 hour, whereas the observed is around 2 hours. So there's probably some room for improvement to really understand what's happening with those suspension. However, when you look for two drug products that have difference in doses and viscosity, the data shows that Tobradex has a slightly lower exposure than the Tobradex ST. So here we have the Tobradex on the left and Tobradex ST on the right, and there is an increase in exposure between two formulations. The model is able to capture that increase in exposure as well. So even though the fit is not perfect, it shows that we are able to predict the direction of clinical exposure between two formulations. We had one more formulation for dexamethasone, Maxidex, and you can see that the model is doing a reasonable job at describing the Cmax and the profile. One thing I want to highlight with this particular slide, that our current biggest limitation is to understand the intersubject variability. Those data were obtained for cataract subjects, patient undergoing cataract surgery, and we have one data point per subject. And you can see how the exposure ranges from 5 to 55 nanograms per milliliter. And therefore, we have strong, very large intersubject variability. And this is one of the limitations to currently validating the models, especially for suspension, because there are other aspects that may limit the predictions. So with that, just a general conclusions that locally acting drug product bioequivalence presents very specific challenges compared to all drug products. PBPK model can support bioequivalence assessment of all of the locally acting drug products, but also very specifically the ophthalmic. The research on PBPK models for ophthalmic formulation is not new by any means, and it started in 2010, and we've been in active collaboration with more than 9 years now with the FDA. And the key aspects we are currently working on is to validate the extrapolation between preclinical and clinical. You will see tomorrow that the early results are very encouraging for solutions, so we are definitely on the right track. And we are always continuing to working on this to, at the end, have a validated tool that can be used by the industry and the regulatory authorities for the assessment of the generic drug products. So at this stage, this whole approach I've been describing is really expected to have a significant impact on ophthalmic generic drug products, but you could also translate those conclusions to other route of administration like dermal, oral cavity, intramuscular, and so on. Thank you very much, and I think we still have a few minutes for questions.
Thank you, Maxime, for the great and informative session and presentation. And we do have a couple of questions here. Where did you find the observed values for cornea and aqueous humor?
This is the hardest part because right now for those projects we can only rely on published information. So for some drug products like the one I will talk about tomorrow, levofloxacin, we were actually pretty happy to have around 7 to 8 studies performed in rabbits and more than 50 studies done in human. So we have a very large data set to somewhat validate the model. So we are only looking at published informations. For other drug products like dexamethasone that I've presented today, you basically saw everything we found in literature, and you can see there were not that many. And this is actually also raising a very important point to the whole model development strategy, that we tend to see a very, very important inter-study variability. And it's pretty normal sometimes to see 2 or 3 times difference in exposure comparing different preclinical studies, even if they have been done in the same lab by the same teams just over the course of like a couple of years. So we have still limitations getting the data. And if you have any data, please come to us and I'm happy to have a discussion because I always want more.
Thank you. And before we have to close this session, I think we have time for one more question. What is the current regulatory opinion on locally acting PBPK models? I know you are not the regulatory, not the part of the regulatory agencies, but from your experiences and communications, in which direction is it heading?
Yes. So, I mean, this is a great question. And of course, I cannot talk to them, but I think the presentation this morning from the person from clinical pharmacology kind of lay out their current vision of model-informed drug development. And it also applies for generics. There is this issue, and just going back to my second slide, the FDA's mission is to encourage the development of generic drug products. So they need to have access to new tools to help them basically assess that the drug product is safe and effective. PPK model is one of the tools they are working on, but I think the fact that they are sponsoring this particular model for 2014, so 8 years now, is kind of a statement from their part that those models are actually more than appreciated and encouraged actually to be used by any sponsor. So maybe at this stage, we do not have like a real case where those models have been used yet for ophthalmic drug products, but there were one for dermal drug product, and the PD endpoint studies was not done because the PBPK model shown that it was not necessary and the local exposure was comparable between the two formulations. So think about the impact PBPK model can have. There's been one case study for dermal, more case study will come, and I think the FDA is very, very keen to at least have a discussion with you about those models and see if the model you're trying to develop is reasonable and the goal you're trying to achieve can be achieved with what you know about your product and the data you have in hand.
Thank you, Maxime. Unfortunately, that's all we have time for in this session. So thank you all for joining us for this presentation. This webinar has been recorded for playback and it will be available on our website and YouTube channel. This concludes the session. Please head over to the lobby and for the next presentation.
Thanks, Vera, and thank you, everyone.